Thank you, Tony. And now that Tony has given a wonderful introduction to the centre, we're going to dive into hearing from some of our PhD researchers. So first up is Elizabeth Phua. So Lizzie's a PhD researcher in the training centre at Monash University, so in Victoria. And she's under the joint supervision of Professor Andrea Robinson and Professor Katja Puss. So in collaboration with her industry partner, CSL, her PhD research encompasses her knowledge in peptide chemistry, pharmacology, and her newly found appreciation for computational chemistry. Lizzie has also gained experience across diverse industries, including testing and analytical laboratories, as well as research and development within the paint and coating industry. Upon completion of her PhD, Lizzie aspires to further her knowledge and research within the pharmaceutical industry with the hope that her work will take her around the world. So let's welcome Lizzie to the stage to hear about some of her research. Just a reminder for anyone who wasn't here for the introduction, we will have the opportunity to chat to Tony and Lizzie one-on-one after Lizzie's talk in breakout rooms. So please hang around for that. Lizzie, yeah, perfect. You are good to go. Just take yourself off mute if you...
Okay, I had to find my screen, but we're good. Cool. Thanks, Tessa, and thanks, Tony. Nice summary of the ARC ITT Centre that all of us PhD research are very lucky to be a part of. And yeah, thanks, Tessa, for the intro. So, as mentioned, my name is Lizzie, from Elizabeth, but Lizzie for short. And yeah, thanks for everyone for tuning in to this seminar today. Tessa mentioned, my PhD is in collaboration with CSL Behring and focuses on machine learning, which is like a type of AI to screen molecules, small peptides as well, for their binding towards different types of plasma proteins. So I'll get right to it. So plasma proteins, if you donate blood in Australia, we do that to the Australian Red Cross or any other similar donation sites. You can also donate your blood plasma, which is this yellow substance that you can see on the yellow vial. Now, more specifically, plasma proteins, they comprise of sugar, salt, water, and plasma proteins. And they help with the transportation of cells, nutrients, drugs, and neurotransmitters around the body, and are very necessary in some therapeutic treatments for those that suffer from trauma, blood loss, immunotherapies, and biomarkers for cancer. Now, as we do have a growing global population, that also means we have an increasing number of people that may suffer from these life-threatening diseases. And pharmaceutical companies like CSL are seeing this rapid growth and need to keep up with the current demands to ensure that there is enough therapies for those that require it. So the current method to actually isolate these proteins from plasma donations is to utilize a method called the cold ethanol fractionation process, which was discovered in the 1960s, way back when. This method has been successful in isolating multiple proteins, but as you can see, multiple proteins such as the proteins you can see on the side of the screen, and we can obtain different proteins from the plasma with really high purities. But there is a current drawback that this flowchart depicts. It's a very lengthy process to isolate different types of plasma proteins, mainly over 5 fractions that require the alteration of different pH, ethanol concentrations, salts, buffer systems as well, and about 28 million litres of human plasma undergoes this process yearly. And if I want to draw your attention to the very last fraction, we obtain albumin, which is one of the most abundant plasma proteins in the human body. So that's about 50% of the total protein content in the blood. And for the comparison's sake, to kind of visualize, only about 1.1% of the materials that you start off with, including the ethanol that you get, the ethanol used to purify the human plasma and the human plasma to obtain only 1.1% of the atom efficiency, which is the albumin. So poor eco-efficiency and low atom efficiency here. So as I mentioned, you can obtain tens of plasma proteins using this method, but the downfalls we need to keep in mind are that even though we can obtain these proteins in high purities, it is a very lengthy process, and those out there, researchers, synthetic chemists, know that the more steps, the lower the yield you get, and as a result of this large fractionation, there's also large wastes that are produced. So the question arises that, is there another way that we can kind of streamline this process to obtain plasma proteins in a one step whilst minimizing waste and improving the yields and product recoveries along the way? So CSL has been looking into one method is, affinity chromatography. So what is affinity chromatography? Now, AC is a technique that heavily relies on a target molecule or our target plasma protein to have high affinity or attraction to a ligand, which is onto a solid material or a stationary material. And all the substituents within the mixture that are passed through this column are washed out whilst retaining their physical and chemical properties too. So we first expose our crude mixture to the affinity medium, and the protein that we desire attaches to the media, and the rest is washed out. And then we can alter the eluent system or the solution that goes through the column to finally obtain the target protein that we desire, and we can then wash out the affinity solid media and hopefully reuse the column. So not only does this process satisfy our goal to obtain a one-step process, it is also with the aim to improve product recovery whilst reusing the material and minimizing waste. So this is where like the green side and the more sustainable process goal that we are aiming to reach will allow us to bypass this cold ethanol fractionation process. So with this in mind, how can we even find the right molecule or an affinity ligand that will bind towards a target plasma protein? So we do need to satisfy some criteria, which is important for our molecule to have high specificity and selectivity towards the target protein, whilst being able to be attached to a stationary phase, and also be resistant to varying conditions and cleaning procedures of the affinity media, and also be resistant to, and be able to be reused without changing its activity or binding affinity. Earlier I showed that nice long list of plasma proteins that are desired. In order to reach our target protein, we need to find said target protein. So we just chose one of those proteins that are in plasma. And we wanted to look at the structure and binding site of the plasma protein, as well as do some literature search to see the structural characteristics and properties of ligands that are known to actually bind towards this target plasma protein. So from our literature search, the target protein that we chose specifically had hydrophobic residues and cationic pockets within the binding site, which allows ligands that have hydrophobic moieties and electronegative domains to form dispersive and electronegative forces or ionic interactions between the binding site as well. What we found in terms of these compounds, well, there was a whole sweep of small molecule drug compounds and short peptide sequences. So from here, we just needed to find all the different ligands or maybe even peptides that were suitable as a ligand. But obviously through literature, there's an abundant out there. So the whole idea of compounds is to either A, develop it or develop a new compound, or B, to just kind of adapt or change some molecules that are already out there. Now, typically, if you were to develop your own compounds, you can use high throughput screening, which allows you to generate lots of data. However, it is resource intensive, and in most of the cases, you only have 1 or 2, maybe 3 successful or hit candidates. And in recent years, researchers have used computational methods to visualize and structurally analyze some of these molecules. However, the drawbacks are you require a lot of computational energy and large volumes of data, and there are some challenges in accuracy. But we can take the positive of both of these and combine experimental and computational methods, which a lot of openly available databases have, by designing and synthesizing candidates that have been either computationally determined or synthesized and experimentally determined. So again, what do we do with all this data? So much out there. Where do we even start? And this is where we can use to help us screen this kind of data. I'm sure everyone out there is aware of artificial intelligence, or AI, and machine learning is a subcategory of AI, which is just the ability of the computer to learn without being explicitly programmed. Now, the basis of a machine learning model is that the ML model will use a data or database to address a problem, and this data is trained using a certain algorithm for it to evaluate a solution. And from here, we can analyze the solution based on its predicted and its true values. And if the errors are really large and really not what we are after, then we go back to the very start to study the problem, go through the cycle again. And if we're very happy with our predicted values, then we can launch the machine learning model. Now, in our case, our problem is that we don't have a molecule that can be used to purify a plasmid protein. So we can utilize the data, which I showed in the previous slide, collated over openly available databases experimentally and computationally to train our machine learning model. Now, it's really crucial to note that the machine learning model must be trained on relevant data. No matter how good your code is or how gold standard the machine learning model is, the moment you put garbage data into your model, you're just going to get garbage on the way out. So this is something to keep in mind when we were building our machine learning model as well. So our machine learning model, we curated a data set based on the openly available data online, and we found that the molecules that bound towards our target plasma protein had hydrophobic and electronegative moieties, and their structure was planar, causing nice pi stacking bonds in between the hydrophobic areas of our target protein binding site. Now, from here, the structure of the compounds can be represented into simplified molecular input line entry strings, or SMILE strings for short, which can then be translated to fingerprints, which the machine can understand, which is pretty much a vector or a matrix of 0, 1s, or bit digits to allow the machine to understand the structure and interpret the compound in a more black and white format than what we see in ChemDraw or drawing it out. So from here, the features are represented and inputted into different linear and non-linear regression models, could be regression models, and we can tune different parameters of these models using different validation techniques as well. Now, from here, we can then expose it to unseen data, and we can have a predicted value. So in our case, the input training data was the structure of our compounds, as well as their binding affinities. And what we found is that from this model that we have, we found very low errors, about 32 nanomolar differences between the predicted and the actual values, or a 2% predicted, a 2% error, which is pretty good. But before we say it was a great or amazing, we wanted to see if this model could predict new sequences that I could input in. So these novel peptide sequences that we input also had the hydrophobic and electronegative moieties as for the data that was being trained at. And we put these novel peptides into our model, and we wanted to see if it could predict it. And unfortunately, we were not that successful. And I had a quick look at the data that we used to train the model and the peptides that we actually put in. What we saw was that the training data had hydrophobic groups that were consistent throughout all the different data points and variable electronegative groups. However, the peptides that I put into the model that were these brand new peptides were the complete opposite. So what we can see here is this clearly shows that our model relies heavily on the data set, and that the data that we use to train the model needs to be relevant to the novel peptides that we put in. So this meant that we needed to further refine our machine learning model. And this was to increase diversity of the data set, so differing hydrophobic and electronegative groups at the same time to obtain different types of binding values along the way, and also try different peptide sequences or even small molecule as well. So from this, we retrained our machine learning model, combining the training data from the databases as well as the data from the novel peptides that I have made. And we followed the same procedure of inputting them into the model, tuning the model, and having a prediction. So what did the results come from this? We were able to increase diversity of the data set. And yes, the errors were reduced, and they were still quite large. What we can say from here is that just introducing 5-6 peptides into the training data set slightly reduced the errors, which gives us a very promising trajectory of what other addition of peptides and other small molecules could have for the machine learning model here as well. So this is ongoing work at the moment, and we can hopefully, if I can present to you all the next time, we can have more of an update and see those errors decreasing as well. So I do see that machine learning has huge potential in combining literature data as well as novel experimental data, and the model being fine-tuned to hopefully achieve the affinity ligand that we can use to purify our plasma protein, and hopefully using affinity chromatography, which allows us to streamline this purification process whilst satisfying the goal of being one step, had high purities and improved recoveries, whilst minimizing waste and allowing us to reuse the media for future purifications as well. So thank you all for tuning in. Just like to thank my supervisors, Professor Andrew Robinson and Professor Kajik Pass, as well as my supervisors and other scientists at CSL Behring. And also wanted to take this opportunity to thank you all. It's been a great opportunity being part of the ITTC Centre, where I've been learning skills both in academia and in industry. I've also gained a lot of experience expressing my research for those that may not have a strong science or science background, or the kind of research jargon as well, and also learning about the different networks that are beyond just academia and how industry works as well. So thank you.